Morning, Professor Carafit here. We're going to talk about what we would have done if we were doing the photosynthesis lab in person so you can understand your lab report more thoroughly. So, the research question for this lab, what is the effective wavelength on photosynthetic rate, or the rate of photosynthesis? Photosynthesis is the process by which plants take carbon dioxide out of the air, make sugars out of it. They do that usually with their leaves. There are tissues in the leaves called mesophyll where chloroplasts are located. Chloroplasts have a double membrane with a thylakoid membrane in the middle. Chlorophyll is embedded in that thylakoid membrane. That's where the light reactions take place. And then in the stroma, the liquid area around the thylakoid membrane is where the Calvin cycle takes place. If you have not watched the lecture video on photosynthesis yet, I would recommend doing that before working on this lab so you have a better grasp of what we're doing here. Now, different colors of light are absorbed differently by leaves. And as we can see here, and there's a figure in your book I recommend you check out to help you figure out what your hypothesis is for this lab. But you'll see that different colors of light are absorbed really well. Some colors, like green and yellow, are not absorbed at all, while red is also absorbed. So we absorb a lot of bluish, kind of light blue. We absorb a lot of red and orange. And those are the colors that the plant absorbs, and these colors down here are the colors that plants reflect. A plant looks green because it reflects green light. So the colors that are absorbed are going to be those colors that drive photosynthesis, right? So the basic equation, 6 carbon dioxides come in, 6 waters, and light energy fuels this endergonic reaction. We're going to produce glucose and some oxygen gas as well. And how could we measure that? Well, we could measure the oxygen gas output, but instead what we're going to do in this lab is measure the CO2 input using a carbon dioxide probe. The way this probe works is it records parts per million carbon dioxide. So out of a million particles of air, how many of them would be carbon dioxide? And that's what the number is. And so this is a Vernier, that's the company that makes it, Vernier carbon dioxide probe. So if we were meeting in person, we would have done a couple mini experiments and got the results and then use those to generate a new hypothesis for the lab report. But here's the basic idea of the hypothesis for your lab report. And you have to do the hypothesis before you make your graph or anything. Otherwise, it's not really science, right? You got to have a hypothesis before you investigate. So, which colors of light will cause the absorbance of the most carbon dioxide due to photosynthesis? And I have red, green, or blue, but we're going to add to that and say white or darkness as well. So which colors of light would cause the most absorbance? So white light is like a positive control, meaning you know it should work. It should cause photosynthesis. And darkness, you're pretty sure shouldn't cause photosynthesis. It's a negative control. So if you put a plant in the dark and it photosynthesizes, you know something's wrong. If you put a plant in white light and it does not photosynthesize, you know something's wrong. So that's what those two are for, is to check what happens in the best scenario possible and the worst scenario possible in terms of photosynthesis. So which colors of light will cause the absorbance of the most carbon dioxide? What do I mean by absorbance of CO2? Well, CO2 is absorbed by the plant. It goes into the stomata, into the plant, and becomes sugar if photosynthesis is happening, right? If photosynthesis is happening, that's what will happen. So what I would do is I would write a hypothesis. Let's pretend we think green light will be the most active photosynthetic color. And then we have an idea of what the others would be. My hypothesis would be something like this. If green light is most absorbed by plants and drives photosynthesis, something like that. Or if green light is the most active color driving photosynthesis, and I expose plants to red, green, blue, white light, and darkness, then green light will show the most absorbance of carbon dioxide, followed by blue, red, white, and darkness. You know, whatever order you think they will appear in terms of which one will absorb the most, which one will absorb the least. You could say something along those lines. That's how one way you could formulate your hypothesis. But you want it to be in the if, and after the if is your explanation as to why you think what you think is going on here, right? Followed by and then you kind of give a real brief explanation of the experiment, and then what the data will look like if you are correct. That's how those are written. If you've forgotten that, go back to the first week of lecture videos. I explain it pretty thoroughly there. Now, if we were doing this in person, we would take our photosynthesis chamber, we would put leaves inside, spinach leaves, and we would lay a piece of colored film like red, blue, or green on top of the chamber. So pretend my plant's in this coffee cup, put it in there, put a piece of green filter paper here. It's basically just a green filter, and then we'd put a white light right on top of that filter, so that white light's hitting the green, going through the green filter, but it's being filtered, so only green light's hitting the plant. And we would let that plant sit in there for 5 minutes, and then we would put the probe in and keep the light on, put the probe in also, and hit record for another 5 minutes, and then it would show a line on the screen. You'll see a video of this in the other video I made. You'll see a line of data. That's how much CO2 is being absorbed per minute. And then we'll get the slope of that line. So we'll get how much CO2 was released per minute by that plant using that slope. And then we would do it again with some different leaves, probably using red light or green or blue light or darkness or white light. And so that's how we set that experiment up. We would do that, hopefully get more than one run so we can do replication and put error bars on it and all that good stuff. So that's what we would do if we were meeting in person. And then at the end of that, we would have a number, how much CO2 was absorbed by this plant per minute. Then what we would do is divide that by the weight of our leaves. Why? Well, when you have 3 spinach leaves, sometimes one's bigger than the others. And then if you're trying to compare your table's data to another table's data, and if they had big leaves and you had small leaves, your data won't match up. But if we correct it by dividing everything by the mass of the leaf tissue present, we can standardize all of the data. So we get parts per million CO2 absorbed per minute per gram of leaf. And that's the ultimate unit that you'll put on your figure. And of course, if we were doing this in class, we would need to be real careful to make sure our spinach leaves were facing the light. So you don't want one upside down or on its side. You want light hitting it directly. We'd have to clean everything carefully. We'd have to do all kinds of things to keep the experiment going cleanly, which you won't really have to worry about for our lab. That's entirely online. But you should include a short materials and methods section where you give the basics of how the experiment was conducted. Here's an example of a CO2 probe sitting in one of those chambers. In this case, rather than a white light, I actually have a red light bulb sitting there. But I found that didn't work as well as using filters and white light. But that's an example of how we would set up the experiment. And so you're going to watch the next video where I actually show you some of that equipment in action and how it works. And then you're going to you've read a Piranha paper, which shows you how to read a scientific paper, and you'll get an idea of how they're written and what's in there and how the wording sounds and things like that. And you're going to write a scientific paper about photosynthesis. So your introduction should contain a lot of information about background on photosynthesis, not background on this experiment, background on how photosynthesis, what it is, why it's important, how it works, and then you'll talk about your experiment. And you'll go from there, oops, sorry, I bumped the microphone. And you'll go from there into your materials and methods, then your results section, which is a written section of what the results look like, followed by a figure and a caption showing the results, and then a discussion section where you tell me why your results happened, tying it back into molecules and light waves and all that stuff, to use that kind of information to explain why your data did what it did. And I provided a handout on how to write a lab report, and I made a video on lab reports and all that to help guide you through this process. All right. I'll see you all soon.